Good morning, everyone. Thank you so much for this introduction, Andy, and thank you so much for the invitation. It's really a pleasure to be here to discuss our most recent work. And so actually what I will focus on today is indeed about a gene called Stathmin-2. And the work I will present is really a close collaboration with the group of Don Cleveland and Martin Marsala at UC San Diego and the group of Kat Lutz at Jack's laboratory. So I think we all agree by now that the discovery of TDP-43 was really a breakthrough in the field with its mislocalization really as a pathological hallmark in the vast majority of amyotrophic lateral sclerosis, both familial and sporadic, and also frontotemporal dementia. And so you can see in this picture that TDP-43 is normally mostly nuclear. We have not talked so much about TDP actually today, so I have not done a lot of introductory slides. I will try to do that here. So TDP-43 is an RNA binding protein, and you can see that it does normally localize mostly in the nucleus, but in affected neurons, it mislocalizes to the cytoplasm, with this really nuclear clearance that is very striking and very early on led to the proposal that loss of nuclear TDP-43 function may contribute to disease. But really one of the issue was that we knew very little about the function of TDP-43 when it was discovered to be important in neurodegeneration. We knew that it was an RNA binding protein and that it was involved in many different steps of RNA life, both in the nucleus and in the cytoplasm. But really only a handful of its RNA targets were known. And it was a very important question to define that, to really determine whether loss of TDP-43 function indeed contribute to disease. And so with my colleague, Magdalene Polyminidou, and many others in the field, actually, we have used genomic approaches, including cryptic that was discussed this morning by Dr. Kiskinis, to identify the RNA binding sites of TDP-43 at a genome-wide level and really determine what are the consequences of losing TDP-43 from the nucleus. And what we found, as many others in the field, is that there are actually hundreds of changes when you lose TDP-43, both expression and splicing changes. And this lower graph, so do I have the, do you see? Yeah, okay. So this lower graph shows you that actually TDP-43 is involved in the regulation of many splicing events, many different types of splicing. But in 2015, the group of Phil Wang really showed an interesting pattern, which is the role for TDP-43 to normally bind in intron and repress the inclusion of specific exons that should not be here, and they are now called cryptic exons. And he found that there were many of them as the other splicing changes. And so we end up in the field with a lot of splicing and expression changes that are really dependent on TDP-43 function. And with this difficulty of really determining if any of these and which ones are actually contributing to disease and could represent therapeutic targets. And so today I will focus on one of these TDP-43 targets and try to explain you why we have actually started to focus on this and why we think it's indeed an important contributor to disease. And the initial work was published back to back with another work independent from Kevin Egan's group at Harvard University. And really the reason why we focused initially on Stathmin-2 is quite simple. When we depleted TDP-43 in human cells and we did RNA-seq to look at all the expression changes, what we saw was that by far Stathmin-2 was the most affected transcript. So it's highly downregulated. And we realized when we looked at RNA-seq data that in the first intron of the gene, you have this peak that appears that is normally not there. And it's there because there is an abnormal inclusion of this of a cryptic exon. And we looked more carefully at this region, and indeed, in this particular region that we call exon 2A, there are some putative TDP-43 binding sites. TDP-43 binds GU repeats, and there is also a putative alternative polyadenylation site. So what we did was to look at ClipSeq data from human cells, and indeed TDP-43 binds exactly in this region on the Stathmin-2 transcript. And what we proposed was normally TDP-43 binds, and this leads to the formation of the full-length Stathmin-2 with normal splicing into these 5 different exons. But when TDP-43 is disrupted, there is an abnormal splicing with the inclusion of this exon 2A that we can detect either by RNA-seq, or you can see here also if we do just extraction of RNA and an RT-PCR, normally you don't have this presence of exon 2A. We also showed that actually there is not a downstream splicing from the exon 2A to exon 2, but rather the usage of a polyadenylation site. And this leads really to the formation of this truncated transcript here, and with the really striking loss of the full-length Stathmin-2 protein in cells depleted with TDP-43. So it's extremely sensitive to the function of TDP-43. So all of this was when we knocked down TDP-43, but then we looked at fibroblasts converted into neurons from individuals with different TDP-43 mutation. And again, we saw that there was a reduction of Stathmin-2 level. But most importantly, we looked at postmortem tissue from sporadic ALS. And these are spinal motor neurons that have been microdissected and submitted to RNA-seq. And you can see that we observe this abnormal cryptic splicing in all of the sporadic patients we have looked at with a reduction of the level of the full length. We can also do just RT-PCR from bulk RNA, and you can see that all the sporadic ALS patients we have looked at have this abnormal transcript. We also see that in C9 or 72 ALS FTD, but importantly not in SOD1 cases where we know that there is no TDP-43 pathology in this familial form. So very consistently, the group from Len Petrocelli has looked at a large cohort of FTD patients and show that actually you can detect this abnormal cryptic splicing in patients who have frontotemporal dementia and TDP-43 proteinopathy, but not in the other forms. We know now that almost 50% of the patients with Alzheimer's disease also have TDP-43 proteinopathy, and we have now evidence that indeed cryptic splicing of Stathmin-2 is abnormal in this subpopulation of Alzheimer's patients. We have looked here also to another cryptic spliced exon that has been recently reported to be important in TDP-43 proteinopathy AG13A. So I've shown you that there is clear evidence that Stathmin-2 is misspliced. It's very sensitive to TDP-43 function, and that was a strong argument for us to focus on that. We were also interested by the known or reported function of this protein, which is very highly expressed in neurons, and actually in the motor neurons, you can see on the top right, they are basically among the top 25 mRNAs that is expressed in motor neurons. Stathmin-2 had also been proposed to be a tubulin dimer binding protein, and it's been shown by Aaron D'Antonio and Jeffrey Milbrandt to play a role in axonal maintenance, in particular in sensory neurons that are encultured. When you knock down Stathmin-2, there is an accelerated degeneration of the axons after axonopathy. However, we really did not know what was the role of this protein, in particular in motor neurons. So we used iPS motor neurons that are grown in a specific microfluidic device where you grow them on one side and they send axon through channels. And then we treat these motor neurons with antisense oligonucleotide to degrade either Stathmin-2 or to degrade TDP-43. Then we perform a mechanic axotomy and we let the neurons recover for 24 hours. And the images you will see in the following slides are from this part of the device where really we see when the iPS motor neurons are treated with a control antisense oligonucleotide, that after 24 hours, they can regrow very nicely. And they actually, this axon are containing abundant Stathmin-2 level, including in the axonal growth cones. However, if we treat these iPS motor neurons with an antisense oligonucleotide that degrades Stathmin-2, we really prevent the ability of the motor neurons to regrow after axotomy. And this can be corrected if we knock down Stathmin-2, but then transduce the neurons with lentivirus that does overexpress Stathmin-2. You see Stathmin-2 is back, and the axons are again able to grow. When we knock down TDP-43, I've shown you this Stathmin-2 protein is basically depleted by that because of the missplicing. And so not surprisingly, these motor neurons are not able to regrow after axotomy. But what was very important and interesting was that when we replace Stathmin-2 using a lentivirus in these motor neurons that do not have TDP-43, we completely rescue this phenotype. And that is important because we know that in these particular motor neurons that do have depletion of TDP-43, there are hundreds of other splicing and expression changes. And that Stathmin-2 is really essential for the ability of the motor neurons to regrow. So this was strong evidence for us to motivate, efforts to restore Stathmin-2 in TDP-43 depleted cells. And there are many ways you can think about it, but one of them is to use antisense oligonucleotides. And we started this collaboration with Ionis Pharmaceuticals. So for the ones who are not very well acquainted to ASOs, I would strongly recommend this review, but very briefly, ASOs can do different things depending on how they are modified chemically. And so one way is that they recognize a specific RNA and they're going to degrade it. And so that is the ASO that I've shown you just before we were using to knock down TDP-43 or to knock down Stathmin-2. There is another way of the ASOs, if you modify them chemically, they can block specific RNA processing events, in particular splicing. And that is really the approach that has been taken for SMA, and that has now led to an FDA-approved drug in this other motor neuron disease. So we tested with Ionis Pharmaceutical many antisense oligonucleotides to block the abnormal splicing of Stathmin-2. And for this, initially we used a neuroblastoma cell line where we had introduced TDP-43 mutation, and this cell line has abnormal accumulation of this truncated Stathmin-2 RNA. And you can see that there is several ASOs that we call rescue ASOs that are able to reduce in a dose-dependent manner the accumulation of this abnormal splicing transcript. And this goes along with the restoration of the full-length Stathmin-2 transcript. We did that then in iPS motor neurons, really using exactly the same paradigm I have defined, where we knock down first TDP-43 with ASOs that degrade the RNA, and then we treat the neurons with Stathmin-2 rescue ASOs that block the splicing. And again, they are able in a dose-dependent manner to block the formation of the truncated transcript and to restore the level of the Stathmin-2 full length. And this is really accompanied by a restoration of the protein level. As you can see in the iPS with TDP-43 antisense oligonucleotide, Stathmin-2 is almost completely lost, and this is rescued. When we treat we use ASOs that block splicing, most importantly, the phenotype is also rescued. When we knock down TDP-43, they cannot regrow after axotomy, and this is corrected after treatment with this rescue ASO. So we have now a way really to rescue, using these ASOs to rescue the level of Stathmin-2 in TDP-43 cells that have loss of TDP-43. But we really wanted to have more information about, is actually Stathmin-2 important for other phenotypes than just axon growth after axotomy. And for that, we didn't know much until recently. I mean, there was these two studies in Drosophila that was supporting that loss of Stathmin is associated with impaired axonal transport, neuromuscular junction degeneration, and paralysis. And very recently, there are two new studies that have looked at deletion of Stathmin-2 in mice and support that indeed this cause motor phenotype with neuromuscular junction defect. And so with catlets, we have taken a similar approach by genetically engineering mice, in particular by generating some knockout Stathmin-2 mice. But this effort, as well as the two efforts cited previously, really delete Stathmin-2 in during embryonic development. And this is really not recapitulating what's happening in disease when TDP-43 pathology occurs in adult central nervous system. So for this, we took two other approaches to test the impact of Stathmin-2 in the adult central nervous system. One was to reduce Stathmin-2 using antisense oligonucleotide in a wild-type mouse, and this is in collaboration with Ionis Pharmaceutical. And the other one is a collaboration with Martin Marcella, who has developed a way of injecting AAV subpially And in this case, we have used an AAV9 that encode an shRNA against the mouse wild-type Stathmin-2. And this allows for a prolonged and efficient knockdown that is local in the spinal cord. So first, our approach was to generate these knockout mice, where we have obtained heterozygous and homozygous mice. They have either 50% or 100% knockout of Stathmin-2. And you can see that the homozygous completely deleted for Stathmin-2 mice are born actually at a normal Mendelian rate, but within a few days, most of them are dying. So ones who do escape and survive, they have a normal survival rate, and we have been able to analyze them. But however, they have a lower body weight, they have a deficit in limb clasping, and they have also a grip strength deficit and rotor deficit. What we see in these mice is that there is a reduction of nerve conduction velocity and also a reduction of the neuromuscular junction innervation. But we do not see motor neuron loss. And so clearly losing Stathmin-2 even completely does not kill your motor neurons. If we look at the spinal motor neuron, at the cell body that is in the anterior horn of the spinal cord, we don't have any loss. However, what we clearly see is that if we look at the motor axons that are in the ventral roots, these axons are smaller than expected, but the number is normal. So as I said, this approach of knocking out Stathmin-2 during embryonic development does not reflect what is happening in disease. So we took this other approach, which was knocking down Stathmin-2 by injection in the ventricles of ASOs. And you can see it's efficiently knocking down in both the cortex and the spinal cord at the RNA and protein level. And again, even if this is a short-term knockdown in these mice after a few weeks, we see that there is a reduction of the nerve conduction velocity, as well as a reduction of the innervation of the neuromuscular junction. But this is only for a few weeks, so we wanted an alternative approach. And we took this approach to inject an AAV that encode an shRNA in the lumbar spinal cord of wild-type mice when they are 1 year of age. So they are otherwise normal animals, and we look at them for up to 8 months. And so you can see that this approach actually allows us to reduce the level of Stathmin-2 in the lumbar spinal cord with a relative preservation and complete preservation in the cervical area. And this is sustained. So 8 months later, we have a strong knockdown of Stathmin-2 to less extent in the lumbar cord, to less extent in the thoracic, and the cervical cord is preserved. This was a QRT-PCR. You can see if we do a FISH, there is a clear loss of Stathmin-2 in the spinal cord in the mice that do express the shRNA. So these mice are fine for 2 months, but then they develop this progressive phenotype with hind limb grip strength. As expected, the forelimbs are not affected because of the local downregulation of Stathmin-2. And we see also a progressive clasping phenotype. Again, as we had seen before, the motor neurons are not dead. So this does not lead to cell body degeneration when we look at the anterior horn. And actually, we don't see microgliosis or astrogliosis in the spinal cord. But we again see this loss of nerve conduction velocity and abnormal neuromuscular junction maintenance. So it's very clear that Stathmin-2 is important for the maintenance of neuromuscular junction. And when we look at the anterior at the ventral roots, these are the motor axon, and we see a very striking loss of the large axons, and with a reduction of the ventral root area, but without a reduction of the number of axons. And electron microscopy showed us this very striking phenotype where we see basically the axons are collapsing with this abnormal myelin sheet, and you can see that the spacing between the neurofilaments that are really the molecules that makes the structure of the axon is collapsed, and this distance is very, very much reduced. Surprisingly, also, Stathmin-2 has been proposed to be involved in microtubules. We don't see a change in the microtubule number, but we see this really strong disorganization of the neurofilament. We have looked biochemically, and in this case, it's spinal cord from Stathmin-2 knockout mice. And what we see actually is that the level of the neurofilament M and neurofilament H are strongly downregulated in the knockout mice at 12 months of age, and as well as the phospho, NFH, and NFM. This is not the case for NFL. So we are still investigating really what this means, but this was a surprising and striking result that Stathmin-2 is really necessary for the structure of the axons and maintenance of the size of the axons in vivo. I'm going to go relatively quickly on this, but it's pretty clear that Stathmin-2 is not specific for motor neurons. I've shown you it's very highly expressed in motor neurons, but what we see when we look at the spinal cord, and this is what is black is stained for Stathmin-2, you see a very strong staining actually in the sensory neurons terminals, where the sensory neurons arrive in the spinal cord. And we know that the IAV injection in the spinal cord leads to retrograde transport, including in the sensory neurons and what we call the dorsal root ganglia, where the cell body of the sensory neurons are. So we looked at that, and indeed, with this method of injecting AAV, we can reduce Stathmin-2 in the sensory neurons. And we see actually that this leads to a reduction of the size of the ventral roots, the dorsal roots, as well as a reduction of the size of the axons. And so this actually loss of Stathmin-2 is associated with sensory deficits in these mice. So I'm just going to summarize what we see when we knock down Stathmin-2 in an otherwise adult normal central nervous system. So we saw abnormal grip strength and clasping. We saw abnormal response to sensory stimuli, reduced nerve conduction velocities that was seen in all the models. However, we don't see motor neuron death, and we don't see glial activation. We see this collapse of the axons of the motor neurons with reduced NFH and NFL levels and phosphorylation. We see reduced axonal diameters of the sensory neurons and disruption of the neuromuscular junction. So really this work strongly supports that Stathmin-2, beyond its role in allowing axon to regrow, have a role in vivo in the maintenance of neuromuscular junction and in the maintenance of the size of the axons. So again, this is further support to develop therapeutics that restore Stathmin-2 in TDP-43 proteinopathies. But for that, we really did not have a good animal model to test, for example, the antisense oligonucleotides that I've shown you before are working in iPSC motor neurons in culture. And we don't have a good model because this exon 2A region is not conserved between mouse and human. And so there came Clotilde and has made even more mouse models. So these are just a few examples. One is to humanize the mouse Stathmin-2 gene. And this works by putting a piece of human region in the mouse gene. I'm not going to describe them because in this particular first mouse model, you need to deplete TDP-43 to see the abnormal splicing. And we wanted another model where we could have constitutive abnormal splicing, so we could really test different therapeutic approach, including the antisense oligonucleotides. And for that, what we decided to do was to insert the human region, but also to mutate the TDP-43 binding sites to really have a constitutive missplicing. And so these are my last few slides. And so what you can see that in these mice that are not only humanized, but have a depletion of the TDP-43 binding sites, the mice that are heterozygous for this are expressing this abnormal truncated Stathmin-2 transcript, and they have 50% downregulation of the full length. So you have a constitutive missplicing of Stathmin-2 without needing to reduce TDP-43. And this is a very important tool and model. to test antisense oligonucleotide. And indeed, we have injected this antisense oligonucleotide I've described just before to rescue the accumulation of the truncated Stathmin-2. And you see that there's a rescue of different using different of these ASOs, and there is a partial restoration of the full-length Stathmin-2 mRNA. You can see here at the protein level also that treating these mice that have downregulation, very low level of Stathmin-2 with this rescue ASO allow us to restore the protein level. And so this, I have to say that we were as a team quite skeptical that we needed these mouse models. And the team at Ionis has really been pushing us, saying we need an in vivo testing of these ASOs, because maybe ASO work in vitro, they work in cells, but once you put them in vivo, they don't all work, and you need to make sure you select the right ASO before you go to clinical trial. And that was, a 2 years work, easy to get this work there and to have evidence and also evidence of which of these ASO work. If I give you this ASO number 3, it was working very well in iPS cells, right? And you can see that this is not the one we are going to move forward with. So sometimes we want to take shortcuts, but we need to be very careful. And I'm glad we could make this mouse model. I hope it's going to help many of the efforts to restore Stathmin-2 levels. I've just very quickly 2 slides to say, we are very excited by the ASO. I think we need other approaches. My group is working on a small molecule screen to look at that small molecule that would restore Stathmin-2. We have also a lot of work ongoing, in particular with Michael Ward, to do CRISPR screens to restore Stathmin-2 level. So just to conclude, I've shown you that alteration of Stathmin-2 is a hallmark in patients with TDP-43 Proteinopathy, including sporadic and familial ALS and FTD. Reduction of Stathmin-2 in adult mice induces progressive motor and sensory deficits with collapse of axons and denervation of neuromuscular junctions. ASO-mediated restoration of Stathmin-2 levels is sufficient to rescue axonal regeneration in TDP-43 deficient iPSC-derived motor neurons. And we have generated novel mouse models that enable in vivo testing of restoration of Stathmin-2 levels as a new therapeutic strategy in ALS and FTD. So as I said, it's highly collaborative, and I think I've cited the collaborator, but clearly Don Cleveland, Martin Marcella, and Cutlass are the other PI on this work. And we have long-term collaboration with many on this side and a lot of support. Thank you so much for your attention.